All right, so I wanted to do a little screencast about the structure of DNA. This should be a review for all of you since we learned about DNA back in the first trimester. So I'm just going to briefly go through this real quick and first remind you that remember DNA was discovered, the structure of DNA was discovered in the 1950s. For a long time, people thought that the genetic information was actually just in your proteins, and you received just a bag of proteins from mom and a bag of proteins from dad, pretty much, and those mixed together to make you. And later experiments proved that that was completely incorrect. So we started to settle on the fact that it must be these nucleic acids like DNA that have all the genetic information. And so the race was on to discover what does DNA actually look like. And we had a bunch of different labs working on that. The closest labs were the lab that Rosalind Franklin was working in, and then a lab run by James Watson and Francis Crick. And so Rosalind Franklin's lab was using this new technology called X-ray crystallography, where they were kind of shooting a bunch of X-rays at the DNA and seeing what patterns emerged. And they saw that there was this helix pattern, but they weren't fully able to figure out what was going on. They were kind of leaning towards maybe some sort of like triple helix, potentially. And she actually showed these pictures to Watson and Crick, and they saw it and immediately were like, oh, we got this now. So they figured out the double helix structure along with the repeating pattern of nucleotides, a sugar phosphate backbone, and they published their results. They got the Nobel Prize. Rosalind Franklin was given no credit by either of them. Huge jerk move. And unfortunately, Rosalind Franklin died before those two revealed just how much help they had gotten from her. And unfortunately, they don't give Nobel Prizes after someone has died. So kind of just got robbed there on that one. So another example of guys in science really just screwing over the ladies in science. So what is DNA? So like I said, it's a nucleic acid. And that first word nucleic probably sounds like something you've heard in the cells called a nucleus. And that's, of course, where DNA is going to be found in the nucleus. And it is made up of building blocks called nucleotides. We learned about other big macromolecules that are found in cells, like proteins, which are just made up of building blocks called amino acids, and carbohydrates, which are just made up of building blocks called monosaccharides or simple sugars, and lipids or fats, which are just made up of building blocks of repeating fatty acids and glycerols. And so when we have DNA, we have these segments of DNA that code for traits, and we call these segments genes. And it's important to realize that all of us have the same genes. That is to say, all of us have a hair color gene, all of us have a eye color gene, but not the same would be what is actually coded for in that gene. And so I'll talk about that in a second. So these nucleotides are made up of three pieces. We have a phosphate group, a sugar, which in the case of DNA is called deoxyribose, hence the name deoxyribonucleic acid. And then we have a nitrogen base. And those nitrogen bases are where all the fun is, because there are four options for these bases in DNA. We have adenine, guanine, thymine, and cytosine. And you can see in the picture there that thymine and cytosine look kind of similar, and adenine and guanine look kind of similar. They have two rings, and thymine and cytosine have one. When we go forward, we will not be calling them by their full name. We're just going to abbreviate them and just call them A, G, T, and C. And when we have the two sides of the DNA, they are always going to pair up in one specific way. Anytime you have an A on one side of the DNA, there's always going to be a T on the other side. And anytime you have a C, there's going to be a G on the other side. If there is anything else not happening with A to T, C to G, it is a mutation and could be a problem. The two sides are held together by hydrogen bonds. We don't want it to be a super strong bond, because we want them to be able to be broken apart when we go to try and make more DNA or when we are trying to make a protein. And when we're talking about these letters, this code, your code is unique to you. We all have the same genes. We don't have the same codes in those genes. So my hair color gene is going to have a different code than your hair color gene. It is going to be a different sequence of A's and T's and C's and G's. Now, people with similar hair color to mine, which is dark brown, is going to have a more similar code than people that have a very different hair color than mine, like very light blonde. But at the end of the day, the genes we share. The codes we don't share. Your code is unique to you. Now I wanted to touch on this word chromosomes, because you can't just use the word chromosome and DNA interchangeably. A chromosome is taking one strand of DNA and kind of just condensing it and coiling it up. If you've ever taken like a string and from the two ends twisted in opposite directions and it gets all knotted up on itself, kind of like that, where you take one long string of DNA and get it nice and condensed into a chromosome. And usually we're only going to see DNA in the chromosome form when the cell is going through mitosis, because we want the DNA to be nice and easily packaged for moving around, such as when we are moving 46 chromosomes towards opposite ends of the cell. But other than that, DNA is usually just in a loose, kind of stringy form. We call it chromatin, actually. Your chromosomes, hence your DNA, are always located in the nucleus of the cell, except for that brief period of time during mitosis. And all of your DNA came from your parents in the form of chromosomes in their sperm and egg cells. You got 23 chromosomes from mom, and you got 23 chromosomes from dad. And we're going to talk about how important that was later in the unit. But this picture kind of shows you how if you take this DNA molecule and you just coil it up, coil it up, coil it up, more and more, coil, 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 chromosome. The shape of the DNA molecule, which was the huge discovery in the 50s, is a double helix. Kind of like if you're on a roller coaster and you're going along and it does that kind of spin you around part, not a loop, but just kind of a corkscrew. And it's sort of like that. You can see we've got the two sides swirling around each other, and then we've got these pieces in the middle here. And so the outside part there is where all those sugars and phosphates are that we were talking about. And they just alternate sugar, phosphate, sugar, phosphate, sugar, phosphate. And then the inside part, that's those nitrogen bases, the A's, the T's, the C's, and the G's. And when we are talking about DNA shape, we sometimes call it a twisted ladder, where the sides of the ladder are those phosphates and sugars, and the steps on the ladder that you actually climb up on, those are the nitrogen bases. And so this picture shows you the nice helix pattern we have here. We've got our A's to T's and C's to G's. So every time I have that green T, I've got that yellow A right across from it, all the way up. Same with the red G's and the orange C's, all the way up and down. So what were the three components of a nucleotide? Hopefully you just said sugar, phosphate, and nitrogen base. And what are the four different types of nucleotides we can have? One's with A, adenine, T, thymine, C, cytosine, and G, guanine. And hopefully burned into your memories now will be, how do they match up? A to T, C to G. That concludes this screencast. Hopefully you are remembering everything there is to know about DNA.